Well, welcome all, and thank you for joining us for this webinar. Um, I'll just spend a few moments introducing the webinar whilst participants arrive, and then I'll introduce our speakers. My name is Peter Charlton, and I am helping to organise this webinar on behalf of the Institute of Physics and Engineering in Medicine, or IPEM. Today's webinar is on the clinical applications of photoplethysmography, and my thanks to Catherine and her colleagues at IPEM who have made this webinar possible, and also to my own host today, the Biomedical Engineering Institute at Kaunas University of Technology, who themselves have a great track record in photoplethysmography, both developing devices and developing new techniques for signal processing. This webinar has been recorded and will be made available to registrants at a later date, and we will do our best to send you a link to view it. Today, we have two talks on clinical applications of photoplethysmography given by Professor John Allen and Professor Panikos Kyriakou. Dr. Daniel Kulin sends his apologies as he is unable to present today due to unforeseen circumstances. John and Panikos are leaders in the field of photoplethysmography, having published extensively on the topic and worked with industrial partners to translate their work towards clinical use. Recently, they edited a new textbook on photoplethysmography together, and I'll shortly put a link to it in the chat. During their talks, we encourage you to contribute questions using the Q&A box at the bottom of the screen. So, it gives me great pleasure to introduce our first speaker, Professor John Allen. John is known to many of us for his seminal review paper, Photoplethysmography and its application in clinical physiological measurement. He has a wide range of relevant experience beyond this paper. In 2001, he founded a unique vascular optics measurement facility at Newcastle's Freeman Hospital for microvascular assessment. In 2020, John started his current role as Professor in Biosensors and Bioinstrumentation at the New Research Centre for Intelligent Healthcare at Coventry University in the UK. John, thank you very much for presenting today. We're looking forward to your talk.
Okay, many thanks, Peter. Thanks to the Physiological Measurement SIG of IPEM and IPEM themselves for giving me this opportunity to speak at this latest PPGA webinar session. I'm John Allen, Prof in Biosensors at Coventry University. That's at the Centre for Intelligent Healthcare there. And I specialise in vascular optics, particularly PPG or photoplethysmography. So good day, everyone. My talk is about the utility of PPG for the assessment of peripheral arterial disease, or PAD. That is really blockages in the leg blood vessels, the arteries, which can be pretty serious and should be assessed in a timely way. But first, though, with a quick bit of background on PPG, there's a way in. PPG, or photoplethysmography, well, it uses light, and it uses this to measure changes beat by beat in, well, we believe blood volume, the blood volume pulse, as we can see on an example there. Very simple technique, and you get some very important information this way about the cardiovascular system. Peter mentioned the 2007 review, and this was in the journal Physiological Measurement, which actually is quite popular for PPG articles. It's done pretty well with citations, 2,000 plus now. So I was very pleased with that. Thank you. Okay, PPG, well, it's been around since the late 1930s, and here's an example sensor shining light on a source, shining light onto tissue, and a photodetector to measure the light that sort of, in a sense, bounces back off the tissue. And that changes with each heartbeat to give you a pulse. Moving into the 1980s, 90s, 2000s, and I spent a lot of my time doing research using analog sort of technology, which was very good, and using PPG sensors based on near infrared mainly. So the sensors really low cost and pretty simple, or simple in theory anyway, an LED and a photodiode. How it attaches to the skin, the tissue, though, is very important and a fundamental component in the whole system. So you can take these analog signals and amplify them and then filter them, as shown on the panel on the right there. And the filtering also is another important parameter with PPG. So, well, PPG now has gone digital, so it's smaller, faster, and smarter. So the sensors have got smaller. Well, they're going to get them in the wearable form, as we can see here, the flexible electronics. The processors have become digital, so you digitize the signal as near to the transducer, the sensor, as possible. It comes up a lot now in your sensors for wearables, such as the watch shown there. So the number of citations has launched off, particularly in recent years, and that's attributed perhaps to wearables, to the modern signal analysis, such as using artificial intelligence. But you can see that that line is going up almost, I guess, exponentially. Oh, for such a simple technique then, well, it is quite complicated when you look at the optics to this and how the light interacts with tissue. It's complex, and there's several processes going on, and many have investigated these processes, and there's a couple of references there. Also, the key factors appear to be involved to do with blood volume changes, the vessel wall movement, the dermal layer movements, and red blood cell orientation, and a mixture of these probably. Wavelength is important, like with all optical measurements, for the reasons shown there. I know that my colleague, Professor Panikos Kyriakou, was co-presenter today. Himself and his team have done an outstanding amount of work in this area, looking at optical considerations for the PPG origin. With my engineering hat on, though, well, I know that PPG, the bottom line, it gives composite information. It gives you information about the microcirculation and the macrocirculation, unlike many other techniques. And that's what I tend to use it for, even if I don't fully understand the physics behind the optics there. But it's important to understand from the science point of view, of course, what it might be measuring. So the PPG review looked at the PPG signal on well, it's got two components, the AC part and the DC part. The DC part is this low frequency change, which is site dependent across the body. The 2007 review noted 3 clusters of areas of interest, A, B, and C, as shown on the slide here, in clinical monitoring, autonomic function, and vascular assessment. And it's vascular assessment I spent most of my career in the NHS looking at. Particularly this condition here called peripheral arterial disease, or PAD. And this affects about 20% of the population over 60. And its sort of incidence goes up with age, and it's a serious problem because it has significant associated cardiovascular mortality and morbidity. And it has a growing burden, well, in the UK alone, on the NHS healthcare system. And so it's often presents as symptoms in the legs first, when things become symptomatic. And this can link in, then it can be associated with blockages in the arteries in the heart and in the neck for heart attack and stroke risk. So it's important really to assess it in a timely way. One technique that's recommended is a technique called ankle brachial pressure index, or ABPI, and that involves taking pressure measurements from the both legs and both arms one at a time, and measuring the ratio between the two. And so if the pressure is about less than 90% of the arms in the feet, then that's diagnostic for PAD. However, the ABPI can be a tricky test to do, and it takes time and with skill. And some patients don't like it. They don't like the cuff part. So that led on to looking at PPG instead, another vascular measurement technique. And here we see a multi-site form of that technology. And this is what I use for my PhD research. A 6-channel system looking from head to foot at the bilateral PPG characteristics in health and in PAD. And so we just knew from the toe pulses that in healthy subjects, you tend to get this nice pulsatile waveform and bilateral similarity. The pulses are identically the same right and left, but they're very similar in normals, shape and timing. We get someone with a blockage in an artery on one side, a unilateral disease, and well, one side's unaffected with the pulse and the other is clearly damped, almost like a sine wave. And so the eye can see this from sort of cheap as chips, sort of £5 sensor technology. And so that led us on then to doing studies and quite a bit of background work in here, developing the system on, a few awards on the way, publications in a hospital-based study, which gave confidence to go further. The technology also produced vascular markers for other cardiovascular diseases, so it has versatility. We wanted to know, though, could it work in primary care rather than hospitals, bringing care closer to home for patients? And so this led us on to applying for some funding to the NIHR, to the I4I Cardiovascular Challenge Award and secured the funding there. And that took us on a 3-year+ journey. I think that journey is still ongoing. And here's the Gantt chart for it. It became pretty complicated. A very wide development team involved of all sorts of specialisms. 4 partner organizations are involved as well. And an excellent project manager to try and link all this together with myself as the chief investigator. Also gained some awards along the way there from the IET. And then the end product is this from us. It's a next stage prototype of the multi-site PPG system, a 4-channel system. And this was funded by the NIHR. And this was, I was the principal investigator, and lead contributors were Dr. Andrew Sims and Dr. Jim Whiteman and Professor Jerry Stansby as the clinical champion. So this was purpose-built and designed for automated use in GP setting, co-created. So it has a potential for game-changer impact. And we've published quite well from this so far, and there's still papers coming out from this, the research and development project. And Newcastle are taking the device commercialization on. We did actually do a clinical trial, and here's some sort of just an overview just to sketch out the results, looking at the performance. If you take the duplex imaging as the gold standard and done by a specialist, the ABPI does pretty well. That should be in the top left-hand corner there if it's actually agreeing with the colour ultrasound duplex scan. We also did other comparators as well, a symptomatic questionnaire, which is the ECQ, chemical evaluation questionnaire, and also the PAD register itself. We looked at this in 2 ways, with a manual analysis, taking through the signals there and manually sort of labeling every beat in the set, and coming up with criteria that were used in the former study in the hospital to to include or exclude recordings and such like. Also, we developed an automatic cluster analysis-based denoising technique as well, because the technique ultimately has to be automated, fully automated. And my good colleague, Dr. Sam Huttart, he was trained to do the PPG manual analysis, and that was for his MRes thesis there. Did a great amount of work. Analysis is actually submitted to a journal, and it's under review, so hence I haven't given the full details away there. We're waiting for the paper to come out. Well, this, though, led us on to another offshoot of an interesting area of work. And we found that with the early prototype PPG system, that signal artifact was higher than expected in the primary care. And noise often gave a false positive. So if the trace was noisy in a healthy individual, it would get actually, with the analysis there, give a damped pulse. And this would sound the alarm bells to say this patient's got PAD, but actually they've got noise in the trace instead. And so it led on to the novel automated cluster analysis-based denoising technique, and that's published. And also led us on to another study, my colleagues and I, and Dr. Sam Huttart, looking at well, what's expected really. What do you recommend in terms of how long you record for, and when to actually throw a recording in the bin where you can't even salvage it. You haven't got a chance of salvaging it, because here with PAD, you tend to be looking at information related to pulse shape. So it should be higher fidelity than perhaps for other types of assessments. Also came out of this was the importance to recognize there's natural variability in the PPG. This can happen over, say, 20 to 30 seconds. So if you take a deep breath, a sort of gasp reflex comes about, and this can last up to 20 to 30 seconds and interrupt the trace from its normal sort of characteristics. And that changes depending where you measure it on the body. Anyway, so this led on to a very interesting area of research anyway, in terms of denoising the PPG. Here's some more research and development opportunities then. Mention, well, PAD sensing, there's great opportunities there with miniaturizations. You can make things incredibly small now, particularly melting it down to its core 2-channel right-left PPG technology, going digital, going small. You can also develop quite disposable, flexible, wearable sensors for PPG as well. But this does raise some issues about repeatability and pro-tissue interface pressure and its attachment, because you do need some pro-tissue interface pressure for reliable recordings, especially when looking at the shape. Denoising of these signals, and also standardization in the measurements. Perhaps this should be application specific, say, have a standard for PAD, arterial disease assessment. Also, another area we've published in recently is looking at artificial intelligence, that's deep learning, to look at spectrogram images of recordings over a couple of minutes in health and also in PAD. And there are differences between these, and it actually can perform reasonably well. We found that the performance from this technique is better, like with a lot of people reported, for higher grade disease, more severe disease, the diagnostic test accuracy will perform better. We also found the low frequency DC effect may be quite important in PAD assessments. And so can we use AI to help us explain this? There's certainly scope for optimization and scope for looking at PPG classification in the cloud to simplify the sensor and the technology that goes with the patient. Also, technology doesn't have to be complicated, such as AI. You can actually use very simple techniques, perhaps to communicate the disease. And this is another area. It's relatively new. How do you actually report a positive test result or a negative test result? And we can use this sort of pulse sounding approach. So you take bilateral toe pulse measurements, and with each heartbeat, you get a sound that goes beep, beep, beep like that, and that implies that the rise time on both sides is normal, and so the operator can learn that pattern, and then get a patient with PAD, and if the rise time is extended on either side, the right or left toe, disease in either leg, then you get a different sound with each heartbeat. And so the operator really has to learn really two rules for that, and it communicates the disease very quickly. So this is potentially what's called cheap as chips. The idea, though, is not just for PAD, and we're linking in with our colleagues at ETH University in Zurich about this, taking this area further forward. Okay, one question that's come up, and I've discussed this with Peter in recent times, is why the PPG pulse often becomes damped in PAD. Well, understanding could help with, say, with PPG PAD data synthesis, for synthesizing new data without having to collect it from patients, for testing algorithms anonymously, for instance, in AI. So, well, does it go damped because of the macro, the big blood vessels, or the microcirculation? Is it the autonomic nervous system? Is it peripheral neuropathy that might be causing the damping? Perhaps a local tissue compensatory vasodilatation might be the reason why it's damped. And also the damping might depend not just on severity, but on location. And that's my impression, that the more distal disease, particularly in the leg, the more impact it will have on the pulse shape and damping. So it's quite a complex picture going on here. And there might be something to do with the connective tissue disease as well. So there's a lot happening that might make a pulse shape become damped. But certainly in PAD cases, most often, but not always, you do get some damping and delay. Okay. But some people don't say, I don't care, the algorithm, the AI algorithm works. It gives it 90% accuracy, we're happy. But it's useful, obviously, from a science point of view to try and understand this better. And it would help us, say, with a data synthesis. So this has led on some recent work with some modeling, looking at using the package MATLAB Simulink. What you can do is take bilateral toe pulses, and these are unilateral PAD cases. So disease down one leg, one side okay. You can put both into the Simulink algorithm and pre-process this. And just by simple low-pass filtering, one-pole filtering, you can actually produce results that actually look pretty much to make sense. So the model actually could be fairly simple to explain the pulse shape, just as a first-order approximation. Actually, we just plotted, say, the time constant of the pill filter here against this, and there is a sort of a general trend down. I don't think it's actually significant because we've only a small sample here. It's just a pilot, really. But it's interesting to take this forward. And this model actually with Simulink allows you to build in the macrocirculation and also the microcirculation, because there could be components from both of these. And if the model was to be advanced further, we could look at the low frequency DC autonomic nervous system input as well. Great scope for modeling. It's also modeling as well. This has come up recently. We have a PhD studentship available, fully funded. The deadline's actually this weekend for those interested in applying. But it's looking at the chest pulse, and particularly some interesting blips I noticed years ago on the trace. That's the upper right-hand one in the blue. You can see which it provides potentially some interesting cardiac timing marks, and it varies spatially across the torso. We also can link this to peripheral pulse as well and do some very fancy signal analysis and modeling across here. Anyway, another interesting area in cardiovascular modeling. There's other ways of looking at the vascular disease detection as well. That's looking at the endothelial tissue. So the inner lining of all your blood vessels, particularly the arteries, have got a layer of this endothelial tissue. That's the macrocirculation, the macrovascular. And in the microcirculation, which is very close to the skin and very accessible and often assessed by PPG, a lot of the tissue there in the capillary is endothelial tissue. And this is supposed to be a hallmark for early cardiovascular disease changes. So it's very important to assess. And you can do this by looking at PPG traces. PPG, not just quiescently with patient resting, but actually in response to an arm pressure cuff challenge. So you cut off blood supply to the arm for 5 minutes or so, and then you quickly release the cuff and look at the flush response, this vasoreactivity, and it's supposed to link to endothelial dysfunction. I've got some traces here actually from a study we did quite a few years ago in systemic sclerosis, which is a connective tissue disease, which actually at the Freeman Hospital in Newcastle I specialised in because it links to Raynaud's. So it's a macrovascular condition. And we applied for funding to the NIHR, the Newcastle BRC, and they funded a PhD studentship. My good colleague, Mrs. Sadafi Iqbal, is now writing up a PhD and got some very interesting results from this. So look at these spectrograms, because the AI techniques love to interpret pictures of things, and these are the flush responses. Of course, the analysis that's been done in this project, I don't want to share too much because we need to publish that, but it's along these lines. So it's got great potential anyway, PPG. So there is some interesting research going on, or perhaps to do. What is the best protocol? These are the questions. What is the best protocol for using PPG to detect PAD? I'm not quite sure. I don't think we know yet. Cuffless blood pressure measurements using AI approaches, perhaps the finger and toe. That should be possible. Interesting thing to look at, perhaps. Give an alternative APPI. Can we reliably detect leg pad from more accessible proximal measurements? I'm not sure. I'd be surprised, but that's something to look at. The light interaction of tissue, pad versus non-pad, very interesting. And how important is the PPG DC, this low frequency component for pad detection? And why does the PPG shape become damped and delayed in disease? And does explainable AI have a role for that specifically? Could the hyperinic flush response AI empowered analysis be used to assess other cardiovascular groups? So you have the test and the measurements, and then you do the AI analysis. And we've got a paper being sent off shortly looking at heart disease, and it does have value. And a question here is, which is more important? Well, vascular aging is important, but is it the big vessels or the small vessels that are more important for cardiovascular or vascular aging? That's a nice, interesting question. And that brings me on to then, well, PAD, because it's age dependent, does link into vascular and aging. And then, well, from Peter's significant involvement in here and leadership in the Vascular Age Network here, well, I've joined this group about a year ago, and a paper has come out from this. I think it's been mentioned at previous webinars, but I think it's a pretty good summary, systematic review of the PPG into vascular aging. Just to point out, yes, it's the World Vascular Aging Week shortly in next month. Okay, and Peter's kindly mentioned the book that Panikos and myself co-edited, but there was significant contributions, of course, from a number of senior authors in the world in this technology. The chapter listing is shown there. So I'm very pleased with that book, and it was a great team effort to produce that. Okay, there's some events just coming up linked to sort of PAD and vascular and things. And well, there was a focus collection that came out recently with an editorial in the Physiological Measurement Journals, about 20 articles, nearly 20 articles there published. There's a link there. The IEEE EMBC meeting, which will be in Glasgow in July, there is a mini symposium on PPG there, looking at innovations in photoplethysmography. That's what I mentioned these, if you don't mind. Okay, so in summary then, well, there's been a resurgence of interest in PPE. It's got great potential. It has utility in vascular applications. PPG can detect PAD. Well, it's not perfect and gives accuracy, say, 80 to 90%, depending on disease severity. Key thing is the core tech can be very cheap. It could be used easy to do. It should be easy to do, and it could be made very accessible. But the measurement protocol is important, and so is denoising. There's no standardization in PPG for pad as yet, as far as I know, but that will come. But there's mysteries remain as well, which is also an interesting challenge. Currently looking at what might cause the pulse to become damped in peripheral arterial disease. Anyway, great opportunities there for new PPG approaches. Okay, well, that's the end of my talk. Thank you very much for listening. Good to share that with you.
Thank you, John, for a very engaging and accessible talk and for signposting us to various resources. I want to kick off the questions for John, and so if you have questions, please do write them in the Q&A. And I'd like to start with a question from Jason K. So John, you have taken an idea from bench to bedside, from the academic setting through to the clinical and commercial setting. And on this note, Jason asks, how did you come up with the innovative idea to use the PPG signal difference between different sites for the detection of PAD?
So that's a good question. Well, my boss was Professor Alan Murray for a couple of decades in Newcastle, and he introduced me to PPG and as a technique. And he was already supervising an MD project with one of the vascular surgeons looking at this pad. So it's probably some of the earliest work in this, and it was very systematically done. It's a very nice MD that's in Newcastle on using bilateral PPG measurements in this, and looking at mainly timing sort of features. I took the work a bit further for my PhD in shape, looking at shape, which is a key feature as well. And so it's in a sense where it's opportunity, isn't it, really? But a lot of innovations that have come from this are from this core, this observation. You often get dissimilarity in PAD, not always, but often.
Thank you. And just a second question from the chat, which Panikos may also be interested to contribute to if you wish. Charlie C asks, could you please talk about the remote PPG technology? And he says he or she says there are PPG-based applications on blood pressure and blood glucose. How reliable are they?
That's a great question. I know from cuffless blood pressure type assessments, they have value for blood pressure measurements, and maybe extra development work to do in certain areas. I couldn't, to be honest, comment on the glucose one, but I think Panikos has been significantly involved in that area.
Well, if I may add very briefly, obviously we all know there is this great enthusiasm now in pushing the PPG in order to use it to measure or detect different biomarkers, and because there is this great unknown where the PPG comes from, people really empirically try to link the PPG with pretty much many things relating to disease. And obviously, we do know that the PPG is affected by vascular mechanical changes, which vascular mechanics relates to blood pressure. And people have seen, and we have seen it in the literature, that certain features from the PPG, whether it's temporal features, first derivative, second derivative, relate to changes in blood pressure. So harnessing the knowledge of features from the PPG, people are trying to build models to be able to measure in cuffless systolic and diastolic blood pressure with some success. A lot of the people at the moment, they were looking at utilizing data sets like the mimic data sets, and they test their models with some really good results. Definitely within the PPG, there is information in relation to blood pressure. The challenges of people developing primarily wearables in this space is to be able to develop those robust models that they would deliver calibration-free blood pressure measurements using the PPG. We are not quite there. We're getting close. But obviously, in order to have a clinical medical device based on PPG for blood pressure, I think there is still a little bit of mileage to go. But nevertheless, the PBG manifests or reflects blood pressure changes, which they can be seen in the different features. In relation to blood glucose, of course, blood glucose have been almost like the holy grail of non-invasive monitoring, and people trying to get it from different directions over the decades. And at the moment, I've seen a little bit of the publications using or utilizing the PBG. Some people, they go for quick solutions, so empirically correlating changes in PBG with blood glucose changes, and it might be some correlation. Again, one of the biggest problems is calibration. The challenge is to be able to have a calibration-free glucose sensor technology, whether it's PBG based or anything else. And this is where a lot of people stumble. They just can't create that consistency and the need for no calibration. I think there is still a lot of room to go on a PBG based glucose sensor, especially you all know glucose absorbs in the higher optical wavelengths, which are not the usual sort of visible NIR. So linking the PBG to glucose, especially the pulsatile PBG, it is still challenging, and there is a lot of scope for more research there.
Thank you both. My thanks to Charlie and Jason for their questions, and John, just to thank you once again for a very interesting talk. Our next speaker is Professor Panikos Kyriakou. Panikos is the Director of the Biomedical Engineering Research Centre at City, University of London. His research focuses on the understanding, development and application of medical instrumentation, biosensors and physiological measurements for the prognosis, diagnosis and treatment of disease. He's published very widely on the topic of photoplethysmography, as shown by having over twice as many publications on the topic as any other author in Web of Science. Panikos, thank you very much for speaking to us today. We're looking forward to your talk.
Thank you, Pete. Thank you so much. Can I confirm that you can see my opening slide?
Yes.
Great, thank you. Well, I'm delighted to be amongst people that they are interested in PBG. Pete, thank you so much for your really admirable initiatives in the world of PBG, initiating webinars and other activities in the field. And also thank you, I want to thank IPEM that is hosting these very interesting webinars. Also, John, thank you so much. You set the scene beautifully. I don't have to say anything about PBG, so I can kick off from for their clinical application. I think as this webinar is on clinical applications of PBG, I think we have to draw the line and we say probably the most successful and most celebrated clinical application of PBG, it is pulse oximetry, which came into clinical practice roughly in the mid-80s, and it dominates pretty much every corner of every health institution, every hospital, and now we see them in the home setting, providing continuous, non-invasive blood oxygen saturation. Maybe some challenges with pulse oximetry, but nevertheless, it is almost like one of the vital signs now that is monitoring every patient that we have in a hospital. So I'm really also delighted that the photoplethysmography research has exploded in the last decade. A lot of people, they are returning back to both basic and applied research in PBG in order to primarily demystify the PBG. We still don't know all the ingredients that make the PBG. So basic research is growing, and obviously the applications of PBG are exponentially growing. And I think this also came in parallel with this meteoric growth of wearable technologies that primarily, most of them, they have at their core the PBG primarily in the visible range. And we see them growing day by day, all the different wearables and what they can do in relation to healthcare and well-being. So in this talk, I will tell you, give you a few slides, just introduce the team. Always in these meetings, I want to create reaching out, obviously, for meeting new people and engaging in collaborations as well. And then I will focus on one particular application where we squeeze the PPG, which is the non-invasive measurement of intracranial pressure, primarily in traumatic brain injury patients. And this is in collaboration with Barts Health, primarily the Royal London Hospital, which is one of the biggest trauma hospitals in the land, and the project is funded by NIHR. So the Research Center of Biomedical Engineering based at City primarily focuses for many, many years. And for me, my PPG baptism started in 1996. And primarily most of my research is around optical techniques and PPG. And therefore, the activities of the center reflect some of my interest and strengths, which is the development of non-invasive sensor technologies, primarily optical, for screening of disease. And of course, cardiovascular disease is at the core of what we do. For me, the dream is to develop this completely non-invasive blood and tissue biochemistry technology, where without a drop of blood, you can measure a lot of your biomarkers in blood. The strengths of the lab is primarily PPG, both basic and applied, developing sensors and wearables. We do a lot of spectroscopy in order to fingerprint different biomarkers within the visible, NIR, and higher wavelengths as well. And of course, a lot of signal processing, machine learning, and artificial intelligence in biosignals. And you can have the best hardware, the best sensor, but it's really how you squeeze the signal, in this case, the PPG, in order to be able to extract vital information in relation to disease and provide clinicians or the users, if it's used at home, on different biomarkers. We are a decent-sized team comprising of academics, collaborators, primarily clinicians, industrial collaborators funding some of our projects, and obviously the backbone of the lab is the array of postdocs and PhDs. A lot of us, we are engineers, even though we have grown over the years, bringing skills from computer science, obviously medicine, biophotonics, physics, but a lot of the core is in medical instruments. So we design, build, and test all the medical devices within our laboratories. We design and customize sensors, optical sensors, of course, depending on the application. They could be transcutaneous sensors placed around the body, but some of them, they could be indwelling, especially for some clinical applications, that they can be nasogastrically, esophageally, or colorectally used in order to utilize light to measure viability of tissues, oxygenation, etc. Within our labs, we have a physiological measurement suite where we can bring volunteers and test our sensor technologies against commercial devices. We can do blood gases and oximetry, and give us a good footprint to be able to evaluate in a more controlled environment some of our technologies that we develop. Of course, we can't do invasive things, and for the invasive or the pathology things, we have to move into a clinical trial, which is conducted into our nearby hospitals, primarily St. Bartholomew's, the Royal London, and Great Ormond Street, if we do pediatrics and neonates. But also we do a lot of in vitro work. So we do a lot of control studies, a lot of spectroscopy to be able to identify new biomarkers, to see where they live and breathe in the optical spectrum. And once we identify those optical fingerprints, we can then develop sensors and test them. In the PPG domain, primarily the three pillars that a lot of research is underpinned in the lab is to understand the relation of PPG with changes in hemodynamics, vascular mechanics, and hemorrheology. And once we work out or we understand the relationship, then we can utilize the PPG for applications both in disease, but also for applications in well-being, primarily wearables. And of course, you must have seen a lot of these applications published in the public domain, and these are some of the projects that we are championing within the lab with applications of PPG and optical sensors. Now, moving on to the core of this talk, which is the utilization of light or PPG in the estimation or the measurement of intracranial pressure. And I don't know how much you know about traumatic brain injury, but it is a massive clinical burden, and patients or people suffer from TBI. This is following accidents, roadside accidents, a blow on the head. And it can happen in many places, from accidents to sports and other activities. And the bullets speak for themselves. I mean, the amount of individuals impacted by TBI per year, it is staggering around the world. The manifestations of TBI, the mortality and the disability of people, very young people, again, is very significant. And of course, the cost of traumatic brain injury is very high across the nation and internationally. Now, if you have a blow on the head and the ambulance picks you up from the roadside, one of the major manifestations that it happens, we get an increase in blood pressure within the scalp. So the pressure is rising, your intracranial pressure is rising, and if it keeps rising and rising, and we all know the scalp, is very rigid, cannot expand, those vessels will probably burst, and we will have bleeding and hematomas within the brain. And the neurosurgeons will only be able to assess what's happening in the brain once the patient arrives in a neurocritical care unit or in a trauma unit within a hospital. And TBI splits into different categories. We have severe TBI, moderate and less moderate TBI. And the neurosurgeons, one of the things they do, not routinely, because it's not an easy thing to do, is to be able to monitor intracranial pressure by placing a pressure transducer in the brain. And in order to do this, and you can see these dramatic images on the screen, they will drill a hole on the scalp, and then they will place, literally stab the brain with a pressure transducer, or it's called an ICP bolt, that it will go down into the brain, and the pressure transducer, it will be able to see changes within the pressure pressures within the brain. So it comes with risks. That's why neurosurgeons are very selective, where they will place an ICP bolt. It can induce hemorrhage, cerebrospinal fluid leakage, or infection. So they don't do it nilly-willy. They have to decide and assess the patient carefully. And if the patient is deteriorating, they might go for an invasive ICP. So as one of the surgeons that is we're working together, it's just me panic, because the invasive ICP, it is probably the most invasive non-therapeutic procedure. Now, if we develop a non-invasive technology that we can continuously and dynamically measure intracranial pressure, that will solve the problem of invasiveness. It could be used to patients beyond severe TBI. And of course, a non-invasive technology that can monitor pressure changes within the brain, it can have overspill in other pathologies, such as migraine, stroke, meningitis, hydrocephalus, especially in children. So the vision within the team, when we've been approached by the neurosurgical teams to see if we can provide solutions, is to develop a completely non-invasive transcutaneous technology. You place it on the forehead, and ho behold, you have this real-time qualitatively and quantitatively measurement of ICP. So we all sat on the drawing board, drawing back from our expertise in PBG and optics, and we do know that we can send light into the brain. We do know that there is a technology available on the market. It's called near-infrared spectroscopy, that it can penetrate the scalp. We can send photons into the brain. Those photons oxygenation of the brain, hence that's what the NIRS does, looking at oxydeoxyhemoglobin. It is a relative change. NIRS doesn't give you absolute measurements, but it's an indication. Clinicians, they can look at trends, and then they can see if somebody's brain is hypoxic, or if there is a problem in relation to perfusion. Since we know and we have this confidence that light can penetrate into the brain, then we thought, well, can we actually look at pulsations of the the PBG coming back, because primarily or exclusively, I should say, NIRS utilizes the DC component or the whole absorbance of the signal in order to make NIRS work. So for us, we develop a completely reflectance optical sensor, and you can see here one of our researchers wearing the sensor. It's almost like a peel and stick, comprises of 4 wavelengths and 2 photodiodes, very lightweight, and these
Panikos, I don't know if you can hear us. We can't hear you at the moment. We'll just give Panikos a moment. Okay, Panikos has just dropped out, which I think will prompt him to rejoin. So we'll just give him a moment to rejoin.
Peter, could we do some of the questions in the interim, if they're applicable, to interrupt?
Let's just see. Panikos has now rejoined.
Oh, yeah.
Panikos,
Okay, perfect.
welcome back. You're muted at the moment, Panikos.
oh, sorry, Pete, can you hear me?
Yes, you just dropped out. Are you happy to share your screen again?
Sorry, I don't know why what happened. Yeah. Can you see my slides?
Not yet. Sometimes there's a delay.
Hold on. I've been talking to myself, I think.
Only for a few moments.
Okay.
Great, it's just starting to share. Yeah, we can see your PowerPoint.
John, Pete, if you're so kind, can you let me where do you think I stopped, where I sort of interrupted?
If you go back, yeah, so it was towards the end of that slide where you were telling us this was a picture of one of your researchers wearing the sensor.
Very well, thank you. Sorry, please let me know if we have an interruption again. So, so we developed the back-end instrumentation to acquire the signals, the photons, pre-process them, and our instrumentation, custom made for this application. It's a modular design, almost like a plug-and-play electronics. We drive the multiplex wavelengths. We acquire the PBGs from 2 photodiodes, a proximal and a distal, enabling the acquisition of signals from the superficial layers of the skin, but also from the brain. The system allows the automatic or the on-demand control of the intensity of the LEDs, and the system is interfaced with a data acquisition system. And this is the research device that we took to clinical trials. This is the hardware at the back. This is the sensor, the wavelengths, the 4 LEDs, the proximal photodiode and the distal photodiode. And this is a typical, we placed this on the head of a volunteer in the lab, and you can see the PBGs from the proximal at the 4 wavelengths, and these are the PBGs from the distal, and really high quality PBGs, very good signal to noise ratio, which obviously brought the confidence that we are good to go to start some of our in vivo studies. Before, though, we went into clinical trial, we had to build an algorithm in order to enable us to understand or identify features within the PBG that they might relate to changes in intracranial pressure. So we've looked at a number of features such as pulse amplitude, upstroke, rise time, early systolic, diastolic decay, pulse width, late diastolic area under the curve, and total backscatter light. And then we develop a model in order to interrogate those features, extract those features, and eventually use those to train the model with ICP. Now, what did we do in the lab before we went into in vivo? we did an in vitro evaluation of this ICP sensor. We developed a phantom technology, and the brain phantom technology had the capability of controlling the intracranial pressure and collecting PBGs, and then monitoring the changes of ICP and PBG. So this is the in vitro model. We're almost having a pulsatile pump, creating the circulation of blood with the expected arteries going into the brain. We develop a brain phantom technology, and this is a vascular brain phantom technology. And on top of the brain phantom, we placed our NICP optical sensor. And we're monitoring PBGs while we were inducing changes in intracranial pressure within this compartment, which is representing the scalp. And this is a photograph of the phantom technology with all the connections around the pulsatile pump. And in here, we have the compartment representing the scalp, the optical sensor on top, and we're driving this pulsatile pump, and we can change the intracranial pressure within this compartment. And here we can see we managed to reproduce the intracranial pressure signal, or coming from the phantom. And then we have been raising the ICP within the phantom from 10 millimeters of mercury all the way to extremely critical ICP values. You know, a typical ICP for a healthy person is between 7 and 15 millimeters of mercury. Anything above 20, 25, it is extreme intracranial pressure. Probably above here, the patient will probably die. So we can see step changes in ICP, and the PBG is riding on every step of the change of ICP. And then we have looked at the behavior of some of these features, and I'm going to show only two. What happens to the amplitude of the PBG with changes in ICP? X-axis ICP, Y-axis PBG amplitude. And when you see that within the range of normal intracranial pressure, up to maybe 15 millimeters of mercury, PBG amplitude is up here. But look at this dramatic drop on the amplitude of the PBG once we are entering abnormal intracranial pressure. It really shoots down. And the same with the upslope of the PBG. Within the normal ICP pressures, we have relatively good values, and then we have this drastic drop of upslope measurements once the pressure goes above 50 millimeters of mercury. So that indicates that some of the features of the PBG are impacted by changes in intracranial pressure, which gave us the way and the confidence to build the model based on some of those features. And then we developed the machine learning model, and we have run the experiment, the protocol 3 times, data set 1 to 3, and the correlation of non-invasive ICP versus ICP on the in vitro experiment, it was extremely high. I know we haven't had a massive number of data, but again, that created enough confidence that actually extraction of features from the PBG and estimation or prediction of ICP, it is very close to the invasive ICP measurements produced by the pressure transducer. And therefore, we went straight to clinical trials, and that was back in January 2020. And we had a lot of start-stops because of COVID. And this is at the critical care unit at the Royal London. And we have evaluated 40 trauma patients where all of them, of course, they had a bolt into the scalp. And we monitored these patients for 2 days continuously. And this is a typical patient, and you can see here the probe stuck on the forehead of this patient, and this patient is wearing an invasive intracranial bolt for measuring the ICP, which is the gold standard. And coming close to the results, towards the end, we can see here the analysis. These are preliminary results on 25 of the 40 patients. And this is the correlation between the non-invasive ICP and the gold standard, very good correlations. We can see here the limits of agreement of, again, these 25 patients between the mean pressure and the difference between NICP and ICP. Again, very good agreement, very good mean. And you can see some results, summarizing results of the rock curves. And again, some of the thresholds at 15, 18, 20, and 25 millimeters of mercury with some very, very good results. So really that created a lot of enthusiasm and a lot of excitement into the project to move into obviously the further stage to see what else we can do in order to bring this to market. And this is really roughly where the project ended by the funder. The project has been presented at the STEM of Britain Awards in 2020, and we have received the gold prize that was given to Thomas, one of the postdocs leading the research at the time. So this is not the end of the project. We are now working towards further optimization of the sensor technology, further optimization of the machine learning algorithms and the instrumentation. We're trying to incorporate the algorithms we develop and we evaluate them offline. We want to incorporate them within the microcontroller so we can measure dynamically and in real time ICP. Of course, this work is collaborative work and is the effort of many people that I would like to acknowledge and sincerely thank. Justin Phillips, one of the original sort of champions of this project, Thomas, the postdoc, that led some of the development of the sensor instrumentation, Chris, the lead of neurocritical care unit at the Royal London, and PhDs from the lab, where they continue enthusiastically their work, Maria, George, and Elisa. Primarily Maria supports the sensor development, phantom development, more advanced phantom development, and George and Elisa helping with data analysis, data science, and machine learning in order to take this project into the next level. So thank you so much.
Thank you, Panicos, for a truly inspirational talk with far-ranging applications beyond TBI. A question from Professor Kirk Shelley. Do you think it would be possible to create a full spectrum PPG? The concept is to create a device capable of scanning through a wide spectrum from ultraviolet to infrared fast enough, i.e. over 25 Hertz, to allow detection of physiologic modulations and substances via numerous, perhaps hundreds or thousands of wavelengths.
Thank you, Peter, and thank you, Kirk. Oh, me and Kirk, we're always on the same wavelengths when we think about PPG, and I absolutely agree with him. You know, we have seen obviously commercial entities utilizing a lot of wavelengths, especially in the visible range, primarily Massimo and their rainbow technologies, to look at different chromoforms of hemoglobin. A lot of people, obviously, in the wearables, they're using 1 or 2 wavelengths to do pulse oximetry. But for those that are spending now enough time in this space, they appreciate the value of having this multiple wavelength technology that it can scan through absorbers and through wavelength from the visible, from the group, from the blue, if you're going after bilirubin, from the blue, green, red, NIR, moving into the other bands that you are entering, lactate, glucose, urea, of course, water bands. And if you can multiplex those wavelengths within a technology, it will employ computational techniques. It will enable you to discriminate dynamically a lot of absorbers within the blood. So how can we do this? Of course, some people might say, let's use monochromatic LEDs. Okay, people have done it. They've done 2, 3, 4, 5, 6, 7. But I think we are entering now the field where there is this acute need. And I know of some commercial entities that are entering this space to develop wearable or transcutaneous spectrometers. Now, for those in the field of spectrophotometry, of course, we can buy a benchtop spectrophotometer from various manufacturers, some of them are bigger than others, but all of them nonetheless are benchtop technologies. Now imagine if we can take down a benchtop from an Ocean Optics or to a Pelkin-Ermer, one of those big working horses of spectroscopy, take them down from a benchtop size into a technology that it can be placed transcutaneously, on a finger clip or on the arm, on the wrist, and enable this sweep of optical wavelengths at high resolution, I think the world is your oyster. Then you can interrogate simultaneously and dynamically a lot of absorbers. And for those, and I know Kirk is a leading clinician, you know, we know when there is a pathology on the patients that are unwell, there is this, it's like an orchestra of the biomarkers, how they play each other. One goes up, one goes down, one moves to the left, one moves to the right. Once you have this global understanding of how all these biomarkers are moving, you will be able to better manage those patients. And I hope Kirk will agree. I fully agree. And I think we're moving into that space.
Thank you, Panikos. Thank you, Kirk Shelley, for the question as well. One final question just to finish with. Haipeng Liu asks, well, he says, thank you for the brilliant presentation. And he asks, is it possible to use PPG to detect the hemodynamics of patient with STA, MCA bypass?
What is STA?
I have to admit, I don't know.
Yeah. Sorry, I'm not, I know enough acronyms, but in principle, without knowing the specifics of the particular acronym, and probably somebody will put it on the chat as we speak, probably the colleague that put the question. The PPG, once you, assuming you place the PPG on a vascular part of the body that enables you the acquisition of good quality high signal to noise PPGs, most definitely those PPGs will manifest changes in your hemodynamics under this big umbrella. And in absence of knowledge of those acronyms, I'm pretty confident we can interrogate the PPG deeply in order to see how the PPG is affected by those changes. It's a very vague answer and very abstract, but I'm very confident the more I see on the PPG, and in the lab now, we've realized that we have to do, and we do a lot of in vitro and PPG phantom technologies to enable us to control within custom-made silicone vessels and tissue and phantoms, what happens to changes in hemodynamics with the PPG or vascular mechanics or even hemorrheological changes in isolation or in combination. I think that capability of now of unpicking the effect of different changes on PPG, it will inform how we can utilize PPG in clinical applications. I'm very happy to take this offline as well.
Thank you very much. And thank you to all for your questions. My apologies for the couple we didn't get round to. I will draw the webinar to a close due to time. And just to recap, today's webinar has been part of a series of webinars on photoplethysmography. And there's a link in the chat for further details of upcoming webinars. So it just remains for me to extend my thanks once again. Thank you, John. Thank you, Panikos, for your excellent and inspiring presentations, and also to thank the Institute of Physics and Engineering in Medicine for hosting the webinars. Thank you all, and I'll draw this webinar to a close.
Thank you, Pete. Thank you all.
Thanks, Peter. Take care. Bye. See you now.